Hello everyone, welcome back to my class videos on Biology for Engineers 21 BE 45. Okay, so this is the common subject for all the fourth semester engineering students. It's a descriptive type questions. Many people ask me what type of question, whether it is objective or not. They are happy to know if I say it is objective, but it is actually descriptive type one. So we need to include as till now in as per the syllabus. If last moment if university changes, then we will discuss the uh, objective type question also. It's not say a big task for me to prepare it. Okay, so here I am explaining the questions, possible questions with the course outcome and RBT level indicated and scheme as well as answer. This is the second topic, nucleic acid. Already the class videos I already covered, and it is available in my YouTube channel, my intuition. You can go through that. And here I am just explaining what are the different CEOs, what are the different RBT level and question as well as the answer scheme. Okay, so these are the four course outcomes which are mentioned in the syllabus. Course outcome means after studying this subject, biology for engineers, what the students will learn and what level questions can be asked. It can be asked even level 6 question cannot be asked, level 5 can also be asked, but usually level 2, level 3 and maximum level 4 questions will be asked in general. Okay, level 5 and level 6 will cover as a project work or any other activity. Things will be covered, the 5 and 6 levels. Okay, so these four course outcomes we need to cover throughout the syllabus. Okay, so first topic, that is the second topic actually, nucleic acid. Actually, it is nucleic acid, DNA vaccines, RNA vaccines, like that. That particular the class it will go. So nucleic acid, two questions, level 4 and level 3 questions I mentioned. Okay, and these levels, by seeing the questions, if you don't have any study material prepared, then it matches that level. Since I prepared the study material based on this level, already I have communicated to you. Now I am giving the question. So that only have to, if you write that covers that particular level. Okay, so analyze the word used, therefore it comes the level 4. That means you have to analyze it. Some topic is given like nucleic acid based vaccines, how you analyze for their effectiveness in curing the diseases, addressing the emerging infectious diseases and improving the public health. Therefore that comes as RBT level 4. CO4, it is connected to CO4 because we have to think critically towards exploring innovative bio-based solution for socially relevant problems. So these are the socially relevant problems, the new diseases, new issues coming. All these are pandemic and are something which are uh, social relevant problems, and we have to think critically towards that. Okay, so with I thought I connected it to that CO4. So both questions are level 4 and level 3 is given. Answer screen, how the answer can be distributed, possibly not exactly. Introduction to nucleic acid, mentioning the two types, 2 marks can be given, that is DNA RNA mentioning and what are generally what are nucleic acid, that introduction. Introduction to DNA vaccines, I can give 1 mark. So 2 plus 1, 3 marks over. Importances of DNA vaccine for curing the rabies disease, that can be given 3 marks. Introduction to RNA vaccine, 1 mark, and importance of RNA vaccine for COVID-19, that covers 3 marks. So this much if you write, it will be answering these two, any of these questions. Okay, so 10 marks each question it given. So answer also given. What are nucleic acid? Nucleic acids are biopolymers, and types of nucleic acid, DNA as well as RNA, and what are the main differences of the bases, the base which are used, and based on that, the nitrogenous base. Based on that, they are, one is DNA, one is RNA. Okay, DNA is a double strand, RNA is a single strand. DNA vaccines, what is the meaning of DNA vaccine, how it is prepared, that is mentioned here. Already it is explained in my class video. Importance of DNA vaccine, it is also given, but it has efficiency, long lasting protection, ease of administration, all these terms. It comes under the importance of vaccine for rabies. 3 marks. Okay. Then RNA vaccines for COVID-19. RNA, what is RNA vaccine? How it is getting, that vaccine is getting prepared. All these things are here, and even some of the company which are not needed actually. This you can omit. We are not asked about any company and all, so you can use only the first paragraph, which is mentioning what are RNA vaccines. Then importance of, I'll delete it and give. Okay. Importance of RNA vaccine for COVID-19 is also mentioned, like high efficiency, rapid development, easy to modify, safe, potential for broader use, like that it is given. Okay, so I think this covers this much. If you write it, it's more than for 10 marks. Okay, so this is actually, when since I gave you the notes, you can prepare. Otherwise you supposed to listen to a lecturer. You only have to prepare your own study material, and you have to write it in exam. Then this, it covers a 10 marks question very easily. Okay, so I think I understood. This is just like spoon feeding. I prepared all the material I'm giving to you, and even I'm deciding the level 4, level 5, level 3 decided. The answer already given to you. Therefore, you find it very easy. What is this level? You cannot understand. But when you prepare, you don't have a study material. Then you are preparing. Then actually it covers that level 3 as well as level 4. It reaches. Okay, so based on some more questions, which can be treated as a normal question, I am giving as assignment as another question, but the same can be asked as a true question. All this covers CO4 only, because it is the same thing, same topic. Therefore, it covers course outcome 4, and the RBT level 3. 2 I didn't mention here. 3 onwards, so 3, 4, 5, 6, I mentioned different questions, possible questions that can also be asked. Okay, as such can be asked, a modified question can be asked. Only the things. So by reading that question, you will come to know what are the possibilities. And level 5 onwards, you don't think that it will be asked. That cannot be asked in exam. That much we cannot put effort. We cannot, a student cannot put in writing the exams. Level 3, up to level 3, and some level 4 questions that you can check. For RNA vaccine also, these are given. Level 3 to level 6 I kept, searched and found and prepared and kept. Okay, so that is for RNA vaccine. Now forensics, that DNA fingerprinting. Two questions again I kept. One level 4 question and other one level 3 question. Actually both questions covering the same meaning actually. Okay, for 10 marks and covering the course outcome, that is CO4. So answer scheme, how the 10 marks, if I am giving, how I will distribute. Introduction to DNA fingerprinting, the meaning of DNA fingerprinting, to that I'm giving 1 mark. The importance of DNA, why DNA only used for DNA is used for forensic fingerprinting application. What are the importance, how accurate it is, all those it covers the importance of DNA. Working of DNA fingerprinting. Okay, so DNA fingerprinting, this is already explained in my previous class. Okay, there is nothing. DNA profile we have to prepare. The importance of DNA in forensic fingerprinting application was not given in my previous note or previous class. Therefore, it is mentioned here. Okay, so this is a new topic for you compared to my previous classes. So some of the importance of DNA, like uniqueness. Each person, except the identical twins. Okay, so that is very clear. Except identical twins, each person they have different DNA. They have their own unique DNA. Okay, even we get our DNA, or so called the genetic information we are getting from our parents. Okay, whatever the information already the parents know, that information already there in our system, biological system. Okay, so their grandparents also, it also be there. So from generations, the knowledge which is they gained and the information they collected, it is coded in the form of in our DNA, and that is already there in our system. That's why we surprise our kids, how they learn quickly, how they know. I is supposed to learn this in the 10th standard. My kid studying the same, or he understood the things in when he is in fifth standard, because already that DNA, that some information already it is there coded. It's very easy for them to analyze. That is my way of explaining. Okay, no, some it's the true fact only. Okay, so but still individual DNA, individual person's DNA, that is unique. Okay, it cannot be compared with their parents or friends anywhere, but it has got a more attachment, more connections to our parents. So if you know a DNA of a kid, a person, we can easily match with their parents. It's very easy to match. It will be matching. So okay, but they are very unique. Individual person's DNA are very unique. Therefore that can be used as a fingerprint, just like our fingerprint. Each person's fingerprint, it is different, just like the zebra horse. Okay, each zebra they having that black white that one, that is unique actually. For us, when seeing, we find it all are equal, but the zebra they identify with that unique pattern. They identify each zebra with that. Same way our fingerprints are unique. Each person has got their own fingerprint. Each person's DNA is also very unique. Okay, so fingerprint is unique, DNA is also unique. Why can't we use only fingerprint then? Fingerprint has got not stability. The second point says stability. Okay, whatever fingerprint is there, while it will rubbing, it will go. But DNA, it is stable. It will remain there for a very long period of time, and it can be very easily collected even from the blood, or saliva, or semen, or hair, any body material. If it is available from that, the DNA can be separated, and we can use it for the study, or use it for the forensic applications. It has got high discriminatory powers. DNA fingerprinting techniques such as a short tandem repeat analysis have high levels of discriminatory powers. These techniques examine specific regions of DNA that contains repetitive sequences, and the variations in the number of repeats provide a unique pattern for each individual. That means it discriminates very very easily. We can identify a person's DNA, and it will not mismatch with others. Old case investigations, we might have seen, see, the case has to be, it has to be given to the Central Bureau of Investigation, like that. If some movies and all we study, and even also we also, any old case which is getting not getting a solution, so we say that it has to be given like that. Why? Because they use these methods, they have all these techniques. They use very old cases also there. If DNA DNA is available of that case, so to analyze, then it can be solved, it can be matched, it can be compared, and it's very easy to solve the old cases. Okay, exonerating and innocent. Exonerating innocent means if some persons may be alleged wrongly, or they given conviction wrongly, means they are identified as a prisoner, but he is not done that actually. So it can be proved by DNA analysis. Therefore innocent people can be saved. Wrongful imprisonment can be avoided by using DNA fingerprinting, and we can create a criminal database with the DNA. If any person do some crime, immediately his DNA has to be identified, and it has to be stored. A profile has to be created, so that next time any cases comes, if he, it is very easy to match and find. Therefore if you have a database available, very easy to solve many unsolved uh, cases. Okay, how it works, this is already explained in my class. The sample collection, amplification, analysis, and comparison. So that's about DNA fingerprinting. Nowadays with the improvements of artificial intelligence, it will be even become more faster. Okay, now some of the assignment questions and questions given here. Level 3. Okay, I'm not reading it, you can read it. I'm giving this material to you. Okay, so limitations, level 4, level 5. Okay, again I'm repeating, up to level 4 you try for studying for exam. Level 5, level 6 questions, that is only for your interest, to know more, not for exam purpose as such. To know more, you can search and find out the answer for this. You can use it as a seminar topic, or you can use it as an assignment topic. Okay, so these cases you can use. So I think I covered it. I hope that my this question answer class videos are also interesting to you or giving some information. Okay, so don't think that only this type of complicated questions comes. Now possible, because now every institutions are becoming standardized. The level of university is improving, therefore level 3 to level 4 questions are always expected. Otherwise earlier stages always we say what, why, that type of questions are there. Recently it is replaced with the explain, brief, enumerate. So these words you might have seen in question paper. Now you may get this type of questions. It may be even identifying, analyzing the question itself takes time. Okay, to understand what actually the author of the question paper he meant, what answer has to be given, that confusion will come. So you have to, that's why I kept this type of questions. Not that the only this type of questions only will be asked, but possibly it can be asked. Okay, to showcase the, or to improve the standard of university, it can be asked. Okay, so that's all. Okay, I think I did my part for the second part of nucleic acid also. Thank you very much for watching. If you are not subscribed, my request to subscribe. If you are watching, please at least watch at least half of the video, though I am giving you the notes. I know that many simply opening that to the going to the description box, downloading the notes and keeping it, switch it off. Okay, do help me by viewing some, giving some view hours to me also. So once again, I thank you.